Hello, everyone, and welcome to Science with an Accent. My name is Fola. I'm a PhD student from the Department of Immunology, University of Manitoba. So today, we're going to be doing something very different because it's the last episode of season 1 of Science with an Accent. Today, we'll be talking about our own research, just to let you know what gets us excited about science every day. Today, I have the usual people I have with me in the studio. Dr. Paul Klassen from dentistry. So, Paul, tell us what you do. What gets you excited in science?
Yeah, thank you very much. And thank you to our listeners for tuning in once again. What gets me excited is trying to translate what we learn in the research to the clinic. I'm, as Fola said, in the dental department, specializing in gum surgery. And so what we read in the textbooks and the journal articles, we like to try to apply it to our patients to give them the best possible treatment. Now, looking at my research that I do in particular, my project is focused on implants and dentures. And so generally, dental implants are a fantastic way to replace missing teeth. They have the amazing ability to replace a single tooth, multiple teeth, or a whole mouth that has lost all of its teeth. In a recent but unpublished study, myself and Dr. John Chernakis from the University of Manitoba looked at implants that are used to support a complete upper denture to replace all the teeth in the upper jaw. In our analysis, we are looking at how the gums around implants play a vital role in preserving the very bone that support the implants. Implants are susceptible to gum disease, just like regular teeth, and so we're looking to better understand how to prevent the gum disease around implants that support a denture. We looked at many factors that can influence the health of an implant, including the sex of the patient, whether or not they smoked, the placement of the implant in the bone, and how quickly the implant was used to support a denture. In a previous study, we found that it was the gum height, also called the gingival cuff height, that had the greatest influence on the implant's health after 1 year. In our new study, we'll be looking at a longer time frame, up to 8 years, to see if the gingival cuff height maintains its protective effect against the test of time. We firmly believe, as every clinician does, that providing the best care for our patients is a fundamental aspect of dentistry, and to do so, we need to continually research and look for new techniques to provide the highest standard of care. The protective role of the gingival cuff is multifold. It increases the space between vulnerable bone to bacteria-laden oral cavity. It provides comfort to allow the patient to clean around the implant with brushes, and it prevents recession that can expose the implant and bone to pathogens from the mouth. This research shows that when planning to do complex treatment for patients that have had the psychological trauma of losing all their teeth, we need to provide adequate amount of gums around the implants to offer the patients the best possible long-term health and comfort.
Amazing finding. So exciting. We're looking forward to seeing more from your study, Paul. Thank you so much. And now we're going to go over to Meha from the College of Pharmacy.
Hi, Fola. Thank you so much. I'm excited to talk about my own research today. Did you know that patients suffering from schizophrenia also have difficulties in distinguish between different textures?
No, I didn't know that.
Yes, there are different players involved in how the brain processes a smooth, silky texture versus a coarse one. For the longest time, neurons have been in the limelight as the major player. However, recent work has shown that star-shaped cells called astrocytes are also extremely important in how brain makes sense of the world around us. My research revolves around studying how astrocytes and neurons communicate and lead to discrimination of different textures at the level of behavior. I have observed that upon disrupting the talk channels between these two cells, there are impairments in sensory processing. The method of communication for astrocytes is called calcium signaling, and upon calcium dysfunction in astrocytes, there are disrupted levels of calcium observed in nearby neuronal cells. Moreover, due to these astrocytic and neuronal calcium impairments, mice are no longer able to differentiate between two fine textures. If we imagine our brain to be a busy traffic intersection, astrocytes would be the traffic lights which guide the flow of information or cars such that it reaches its destination. Now, if the traffic light is not functioning properly, information doesn't know where to turn and how to reach its destination. Similarly, when astrocytes are no longer functioning normally and gatekeeping the flow of information, it leads to utter chaos and sensory deficits as observed in my research as well. So this research deepens our understanding of the way astrocytes and neurons communicate with each other and further can potentially open gateways to study astrocytes as therapeutic targets for conditions such as schizophrenia.
Oh, wow, that's really amazing. It's good to know all these things happening in our brains. Thank you, and we wish you well with your research. So, my research is kind of more, technical because I deal a lot with proteins, what goes on inside a cell. So, just imagine inside a cell, and then what is going on, all the networks and signals going inside. That's what I'm focusing on. It is complicated, but I'll try to break it down. So my research is focused on how, the normal function of a protein called APDS, what happens when, the protein loses its function. And it's common in a disease called APDS, where a defect in this protein makes people susceptible to recurrent infections. So when the protein is bad, people start to constantly get the flu, get the cough, get cold all year round. Now, this disease usually starts from a young age, and as they grow older, they may experience further lung damage and even autoimmune disease, which is when the body's immune system tries to fight itself. Now, previous studies from other scientists have shown that this protein is abundant in immune cells, especially the ones called B cells, which make all our antibodies in response to an infection. So studying these particular cells in the context of this disease has been the focus of my research. So for us, we're studying this disease by using mouse models that have been made to genetically, reproduce this disease in B cells. And we have looked at aspects of the airway, such as lung function and structure. We'll also be looking at, immune cells in the lungs and other tissues like the spleen, which is an important immune or something we like to call lymphoid organ. So in addition, we will, do experimental experiments to kind of trigger airway inflammation in this model and just compare them to normal mice with no defect in the lungs and see how it works. So far, our preliminary experiments have brought out 3 key findings. The first one is that having this, hyperactive defect in this protein in B cells can alter the structure of the lungs, making the lungs really stiff, and therefore we cannot breathe normally. So that is very interesting what we found, that this protein is important in either making the lungs have its normal structure. The second finding we've seen is that, in the lungs of this mice, there is an increased population of B cells that make a very unique suppressive product called IL-10. And IL-10 will hinder the function of other bystander cells in the lungs which are playing around and doing their function. So in other words, when you have a dysregulation of this protein, it will affect other cells who are trying to do their normal jobs. In other ways, you also compromise in the lungs. And lastly, we found that when we compare normal mice with this mice with a defect, we see that the lungs show the hallmark of being more inflamed and having more signs of lung damage, such as having excess of mucus and collagen being deposited in the airways when we try to trigger asthma in this mice. So clearly, a defect in this protein will just make the lungs of this mice, respond in a way that shows more inflammation. So this can kind of explain why in patients with this defect, we're seeing a lot of this recurrent airway, disease. Their lungs on a good day are not even good. You have expansion of, suppressive cells, and you see the lungs being more inflamed. So this is just, 3 key findings we're finding out so far in our study. And I can summarize from my study so far that, the normal function of PI3K protein is B cells is very important to the normal functioning of the lungs. But like I said, we're still doing more studies to be able to make a full conclusion. And I hope to be able to give you such exciting findings when I know more. So everyone, it's been fun. We've enjoyed, being able to talk about research in general and talk about research. We're gradually coming to the end, and we're excited for the next season where we're going to have Science with an Accent. And Mary, do you have any last words for our audience?
For sure, Fola, I would like to thank all the audience members for being such kind and interested listeners, and also reaching us at our email IDs and socials with all their thoughts and feedback and questions. It's been very good so far, and I'm very excited to be back for season 2 and share some more exciting research in the future.
Amazing. Paul, any last words?
Yeah, I'd just like to thank all our listeners once again. and just keep an open mind and keep learning.
So everyone, it's goodbye from me and my team, and everyone remember that
Science has an accent.